Okay, we're going to observe and identify some cell organelles here. We have 6 different pre-made slides. We have 2 plant, 2 animal, and we have 2 unicellular organisms. So you'll notice that these plant and animal cells, you can kind of see them on this slide is because they're stained. And there's a reason that they are stained. We're going to talk about that in the next part of this section. So when you look at these on the oral smear, they're going to look purple, which are cheek cells, but cheek cells really aren't purple in real life. So looking at these 2, the unicellular organisms, they're going to be the most difficult to see under the microscope. So I'm going to talk about how you're going to be able to see them the best. So I'm going to start with this paramecium. I'm going to make sure that my microscope is ready. I'm on low power and my stage is completely lowered. Turn on my light and I'm going to get started. Put my eye to the eyepiece, move the coarse adjuster toward me until something comes into view, until I say, whoa, wow, that's cool. If I do not say whoa, remember, I don't see it. So I want to make sure that whatever I see is in the center of my field of view, remembering that your field of view is what you can see through the microscope. So you want your organism to be right there, right in that middle. Once it's there, I'm not going to move anything here with my coarse or fine adjuster. I'm just going to flip it to medium power. When I flip it to medium power, I'm going to need to move that coarse adjuster just a bit. And I'm like, oh, that's even cooler. I can see it in much more detail now. Make sure that it is centered one more time. Make sure I have to move my slide just a tad. And then I can flip it to high power, making sure that I'm not going to use my coarse adjuster at all on high power, but I can use my fine adjuster. And when I do that, I can definitely see that paramecium in quite a bit of detail. So what I need to do is I need to draw that organism. And I'm going to draw whatever I see in my field of view, just like we did back with the letter E. And then once I draw that, what I see in the field of view, that's exactly what I see, I need to label the organelles. And the organelles that you're probably going to see are cell wall, cell membrane, cytoplasm, and nucleus. You're not going to see any of the other ones, probably because they are so small within that cell and we don't have a microscope that goes that much into detail. But speaking of detail, you're probably going to want to write down that I'm looking and drawing this at 400 times magnification. And then I'm also going to label my diagram. So right here, I'm going to label something, and right here, I'm going to label something. And even probably right here, I'm going to label something. So there are 3 things that I'm going to label on this diagram. Remembering that these 3 things that I see are one of the organelles in that list above. You're going to do the same thing. You're going to draw, and label each of the other 5 cells.